Welcome back to our final lesson in genetics and biotechnology. This is on applications of biotechnology. And of course, students will learn what that big fancy word means in this lesson. And they will learn that we are now able to insert gene sequences directly into plasmids. Of course, students will have to learn what plasmids are, and that's bits of circular DNA that's found in bacteria. And in this type of biotechnology, we're able to use the work of the bacteria to produce something for us with a gene that we've inserted into the plasmid. And in this case, we will learn how scientists insert a gene for producing insulin into a plasmid and then reinsert that plasmid back into the bacteria. And in this way, the bacteria will produce the insulin. Isn't that fantastic? A little bit of background about your prep for this. You will want to maybe run your gene sequence off maybe on white paper and your plasmid sequence off on blue paper. And you'll see that in your teacher's edition. You have a page to copy. You'll see a whole bunch of base pairs. You'll see a gene sequence page and a plasmid sequence page. And those are two different pages. In this picture, this is the plasmid sequence run off on blue. And this is the gene sequence taped together. It's run off on white. There's part of the gene sequence is shaded, and that's the part of the gene that we're interested in, the part that will code for developing insulin. But scientists will take the parts surrounding that part of interest as well. So follow the protocol in the book, and students will see how this works. There are some fantastic videos. Students will learn about the restriction enzymes that are able to cut DNA at very, very specific parts of a code and leave sticky ends. And then the gene sequence is introduced, and the restriction enzymes are able to place that part of the gene right there into the plasmid, based on matching it up using the rules for base pairing that students learned earlier. That's called ligation. In our classroom, you'll use tape, and in the lab, they use enzymes. Interesting note that the bacteria that scientists are now using to produce insulin is E. coli. Students may have heard of E. coli in a less than pleasant situation. This brings us to the end of our Hudson Alpha unit on genetics and biotechnology. We encourage you to continue to use the words of the cross-cutting concepts and scientific and engineering practices throughout all of your teaching, including these Hudson Alpha units, and hope you've enjoyed this a lot. And we'll see you back for a few minutes about Lesson 11.